Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome to the course of structural biology. We are going through the module of structure based drug discovery and today is the last class of the course. So, in the previous class we talk about what is a good drug candidate, what are the qualities it should have, good activity, selectivity on the right target, elicit desired biological response, but at the same time it should have admetox. So, the definition of a good drug candidate is a drug candidate suitable for clinical testing, is expected to bind selectively to the receptor, elicit the desired functional response. In addition to have adequate bioavailability and biodistribution to elicit the desired responses in animals and humans, and it must also pass formal toxicity evaluation in animals. And a good amount of current research effort is vested on developing in silico admetox in drug designing, because as I told, the entire drug design cycle which is now working through difficult drug targets, the problem is more and more difficult targets, more and more taken cares, which gets more and more longer. So, more money, more time, and that makes the value of the drug a lot higher. So, these are all problem. So, to solve the problem, one of the best way is to replace the experimental procedures with computation. With computation means with computational algorithms, with computation in prediction of the structure, with computation in training, pharmacophore modeling, QSR, docking and all of them. But one of the major aspect is, after doing all of them, when you think that you already get a successful drug, it could be that the molecule is not druggable. So, druggability is become a very important issue which could be very well solved by replacing a in silico admetox process. And by after discussing about in silico admetox, I have introduced the case study for combating antimicrobial resistance targeting enzyme beta lactamase applying theranostic strategies. I talk about the DTMS model. The DTMS model talk about diagnostics, the D is coming from there, the therapeutics, the T is coming from there, mapping, the M is coming from there, and surveillance, the S is coming from there. So, a lot of things you could do, but what we focus here is therapeutics, and I talk about the challenge in therapeutics. In previous days, when you were a drug designer of 1930s, 40s, 50s, 60s, 70s, you have to design a new drug, means you have to design a new drug. But now, when you are a drug designer of current days, you have to design new drug targeting the intrinsic property of the enzyme. I explained what is intrinsic property, and to get into the intrinsic property, what is needed is enzyme characterization, identifying newly emergent mutants in case of drug resistance. And to do that, the hypothesis was every beta lactamase are different with individualistic uniquenesses. So, what is the solution? We have to study each and every beta lactamase coming out from any organism, and every organism their variants, which is impossible, because there are thousands of genes, even 1000 is an understatement. We need cloning, overexpression, characterization of all of them. So, we have how to solve them. The field of drug development therapeutics in antimicrobial therapeutics development, now adopting an interdisciplinary approach combining microbiological screening with computational and experimental techniques, as I talked about. And to explain the point where you could take classes after classes to explain those things, but majorly I divide it into 4 parts. One, the microbiology part, which contains the sample collection, identification and genetic characterization of new gene and mutants, novel gene, novel mutants, where you also include, we have talked about gene, but genomics, metagenomics, transcriptomics and related NGS analysis are also included. On the other hand, in computation, again there are a lot of things, but in broad, develop computational database, in silico work, then develop training set, then do machine learning based predictions, machine learning based group developments, classifications and all of them, and then go to cloning, overexpression, purification, enzyme characterization and then drug designing. So, here you have microbiology, computation, biophysics, biochemistry, drug development. And from this course, as you are in the last class, I hope you have followed the course and you realize that in between there are many others. So, this is a true interdisciplinary where everything is connected. You could go in that way, you could go in that way, you could take both the ways. So, a lot of complicated steps, complicated strategies we need, because drug resistance is, antibiotic drug resistance is a big problem. And as I always talk about, continuing this course, standing on the COVID-19 era, it would not be difficult to even to a child to understand the seriousness of drug resistance. And if a viral drug resistance, if the attack of a virus, one virus could make the whole world kind of in a situation where we have nothing to do, nothing to combat. So, you think so many bacteria, what is the potential they have? With that, I have talked about those interdisciplinary approaches, complicated things, but I take the one simple, relatively simpler case studies to develop anti-tuberculosis drug development by inhibiting the beta lactamase with novel boronate transition state analogues, which are called BATSA, boronate transition state analogues. So, BATSA is boric acid based transition state analogues. So, there is a lot to talk about TB, but I just talk about significant points. In 2011, 8.7 million people fell ill with TB. 1/3 of the world's population is latently infected with Mycobacterium tuberculosis. New infections occurring at a rate of about 1 per second to the whole world. Standard TB chemotherapeutics takes around 6 months. So, you are affected with TB, it takes 6 months to be recovered. More importantly, it is an infectious disease. So, you cannot, and it is airborne. So, if you are in the population, in the crowd may get infected. So, drug resistance is another problem in tuberculosis. increasing threat of MDR and XDR TV. What I was talking about, the resistance, MDR is multiple drug resistance and XDR is extensive drug resistance. So, whereas in one side it is difficult to combat tuberculosis, on the other hand it is becoming even more complicated than what we are looking at with the emergence of multiple drug resistance and extensive drug resistance tuberculosis. So, it is very important to get new therapeutics to combat against this deadly pathogen. Plus C, the beta lactamase, the serine based beta lactamase of tuberculosis has been identified as a major resistance factor. So, now you know about beta lactamase, we talk about beta lactams are the drug, beta lactams. An example is penicillin. We talk about innovation of penicillin by Alexander Fleming, who invented it in 1928 and got Nobel Prize in 1945, and when he got Nobel Prize he very clearly alert the world about his concern regarding drug resistance, but that time because of money, power and the magic bullet, beta lactams are considered as magic bullet because it is one drug which could kill any organism. So, people did not stop using them, as a result emergence of drug resistance happens, and drug resistance comes with beta lactamases. So, what is beta lactamase? If you remember, beta lactam have is typical 4-membered moiety with other groups. The enzyme transpeptidase attacks here and form a OC. So, if you see there is a carbon, so this carbon involved a covalent bond. This is a very rare case where an enzyme develop a covalent bond with the substrate, but because of that the enzyme transpeptidase become inactivated and transpeptidase was involved in cross linking the peptidoglycan layer, if you remember. What is the effect of cross linking of peptidoglycan layer? If you look at, if you have threads, you know, individual threads are very easy to tear out, but if they are cross linked, by cross linking they would form the net type environment and all of them are together, now they are much more powerful. The enzyme which take part in the cross linking is transpeptidase. So, beta lactam, that is penicillin, by inactivating the enzyme transpeptidase makes the bacteria unable to develop the cross linking of the outer cell layer. As a result, the bacterial outer layer become really weak, and when it goes through different environment with high pressure, with water going into it, burst and bacteria died. Now, bacteria comes up with a enzyme which is called beta lactamase. This have the step, but it have another step, this is called acylation. They have a step deacylation. There are two type of, one is serine beta lactamase, another is metal beta lactamase. BLASI is the serine beta lactamase belonging to class A, which is present in Mycobacterium tuberculosis and identified as a major resistant factor because it do not allow any of the beta lactams to work. So, is BLASI a possible pharmacological target to overcome beta lactam resistance in Mycobacterium tuberculosis? So, what we are thinking, as I told, there are two steps, one is acylation and another is deacylation. Acylation is taken by or made by transpeptidase, whereas beta lactamase, it goes through both the step acylation and deacylation. Through deacylation it makes the drug ineffective. The concept is somehow if we inhibit the deacylation step, totally would be great, but at least partially, then it would take time to make the drug, the penicillin drug, ineffective. It will take time. So, first the small molecule go and inactivate the beta lactamase and it would be added with, let us say, ampicillin, penicillin and all this. So, it would be a combination of the inhibitor molecule along with the classic beta lactams. So, we are looking for small molecules which could go and at least partially stop the deacylation step. So, that is what the strategy. Why BLASI is an attractive target? Significant experience with beta lactams and resistance mechanism. So, as I told, in 1928 penicillin was invented, in 1940 the drug came in the market and it says that around 1940 resistance gene was also there. So, think about from 1940 to 2021, which is 81 years. Around the world this gene is mutated, make variants by any way, and because this gene or this protein product, the gene product is responsible to make inactivate a group of drugs which is prescribed by the doctors. More than 75% of antibiotic is beta lactams. So, definitely there are significant research effort to understand. So, we have previous data and as we have discussed, now we are continuously going through innovative computational algorithm, new computational power. So, this is one of the good target. It belongs to a class of beta lactamase for which inhibitors are effective. Inhibitor resistant phenotypes are possible. So, once you get the drug to be activated on the wild type beta lactamase BLASI. What is wild type BLASI? The original BLASI collected from tuberculosis. You will also screen it for the resistant ones and you could make, as we have discussed, initially you screen, then you get the lead, and here the good part is you know that this is a good lead. So, once you modify the lead and get success against the resistant ones, it would be contributive significantly towards development of new generation therapeutics. But at the same time, as I told repeatedly, you have to remember it in your mind, the fact that we are designing this drug in the current era where a little mistake will help bringing the resistance. So, it is not you need a potent inhibitor of beta lactamase, it is also you need to take considerable consideration strategies so that the bacteria or the nature cannot fight back to develop a resistant variant against the drug you have optimized. As I told, beta lactamase are vividly studied. So, you will see that you will get structure in for different class A beta lactamase, like here I have given some examples. Beta lactamase from Acinetobacter baumannii, Mycobacterium tuberculosis itself have its structure, the Blasi have its structure. The first structure of Blasi was 2-GDN, which was solved by Saccatini group. Then there are structures from Bacillus licheniformis, Escherichia coli, Klebsiella pneumoniae, Bacillus cereus, Pseudomonas aeruginosa, Thermus thermophilus, and many more. Also, if you take a close look on the tuberculosis beta lactamase structure, as I told, the EPO structure, the 2-GDN1, and then structures we got NXL 104, which is avibactam bound, cefamendol bound, cefuroxime bound, cephalotin bound, ampicillin bound, mesillium bound, and tebipenem bound. So, existence of different structures of the complex will be, and they are all high resolution structures. So, they are of same proteins, but if you compare them, you see little deviations, you see binding residues, you see loop movements. All of those informations are extremely critical towards the designing of the drugs. There are, as I told, lots of drugs, new drugs developed, drugs tried, like carbapenems, avibactam, penems, mechanism based inhibitors, definitely different generation of penicillins, different generation of cephalosporins. So, all those drugs are characterized, giving us more introspective about the enzyme Blasi from tuberculosis. And with those knowledge, this case study focused on transition state inhibitors. What is transition state inhibitor? I have explained it already. You have A bond B reacting to C bond D will make A bond D and B bond C. So, to do that, what is needed? First, this bond have to be broken, then this bond have to be broken, then this bond, you could write it in that way, would be better. This bond have to be formed and this bond have to be formed. So, this is partial bond breaking, partial bond breaking AB, partial bond breaking DC, partial bond forming AD, partial bond forming BC. This state where all the bonds are in the state of forming and breaking, this is called transition state. And people like us who understand enzyme, who study enzyme, their chemistry, physics, one of the most popular and established theory is nature have developed enzyme to stabilize the transition state of a small molecule substrate. What I say? Nature, Mother Nature I mean, develop or design enzymes in such a way so that they could stabilize the transition state, because by stabilizing the transition state they could lower the activation barrier needed to perform a reaction, and that is the main reason a biocatalyst, an enzyme, could perform a reaction at normal physiological temperature and pressure and other conditions. So, if you understand the hypothesis, what is the outcome is the enzyme bind to the, it is not designed to bind the substrate, it is not designed to bind the product, rather than the intrinsic property is to bind transition state. This is the most critical part. Transition state needed to be stabilized, and whatever needed to perform the reaction is to break the bond and form the bond. So, what pushes it is the enzyme by properly allocating its amino acids so that it could help the optimized position in terms of geometry, in terms of energy for a transformation. So, binding to transition state is the intrinsic property of the enzyme. So, what? Remember the story I told in the last class. If you understand someone's intrinsic property, you will influence the person hitting on that. And also, if you say that, if you try to understand that, you also understand now when we have machine learning algorithms growing anyway working in biology, if you start getting proper data and analyze them, you get a pattern. The pattern, if you now try to continue or connect your dots, getting the pattern is actually getting the trend, which is getting the intrinsic property. For example, in a very basic way, if I am in love with cricket, so if you try me taking to the ground, I will love to play cricket, and if there is not availability of cricket arrangement, I would still love to play something related to cricket. Like if you see most of the cricketers like Kapil Dev, like Ajay Jadeja, Ravi Shastri, they are good golfer, because it is not surprising actually, because it is the same type of game. So, you get the pattern, that is what I am saying. When we get the pattern using machine learning algorithm, so now we design it, and here I am talking about the inhibitor designing, which are BATSAs, boronic acid base transition state analogues. It also called Butts's boronic acid base transition state inhibitors. So, look at the core concept now. As you now understand, our goal is to get the transition state, because getting transition state means you get the connection of intrinsic binding to the enzyme, and you make those analogues, they will bind to the enzyme much more tightly. You understand? So, what we did, we look at the reaction pattern of cefotaxime. This is the enzyme where the hydroxyl group of the serine, the lysine takes the proton and it attacks, and it forms a bond like that. Now, if you have a carbon, have 4 bonds. So, it would show its 4 bonds. Now, if you take a boron would mimic that transition state by introducing the 4 bonds, because one bond is, boron have 3 bonds, but here one bond would be forming and one bond would be breaking. So, 4 bond which is mimicking the carbon. So, by introducing a boronic acid, we actually make a mimic of the transition state. Now, you might ask a very interesting question. Transition state is a state where a bond is forming and a bond is breaking. Both of them do not have any physical existence. How we could see them? It is a extremely well, or I would say extremely like important question, but the answer is we could do it by setting up the QM/MM hybrid method of simulation. I am coming into that. So, first of all, how we have designed the Butts's? We have 3 designing logic behind the development of the compound. First, I have already discussed, we introduce the central boronate which interact with the catalytic serine hydroxyl. So, introduction of these is the designing logic 1. The designing logic 2 is to introduce the R1 group. Here we play safe, but it is actually not safe, because I mean it is safe, but we have a huge database of existing beta lactams. So, we take the best beta lactam moieties who are interacting with the beta lactamase in a excellent way to design that. So, the designing logic 2 is R1 group resembling beta lactam residue, allowing for specific interaction. We already know some of the beta lactam drugs which have excellent binding. So, we take their moiety. You understand that in designing logic 2 we play safe. In designing logic 3 we are very adventurous. So, we put any group, any possible hydroxyl, amino, methyl, chloride like halides, phenyl group, everything. So, this is the designing logic 1, 2 and 3 logics, introducing the central boronates R1 group which are already tested, marketed, approved beta lactams, the best moiety of theirs, and designing logic 3 is introduce anything here. We take, as I told, penicillin series, cephalothin series, ceftazidime series and cefoperazone series. So, these are the moieties we have taken. By taking this moiety, we do the changes we talked about, and that help us. So, we make the library. It contains around 1 lakh compounds. Then we develop the parameters of all of them. We perform docking based virtual screening. What is docking based virtual screening? What we did here, we have known beta lactams. So, we use those beta lactams, let us say beta lactam A, beta lactam B, beta lactam C, beta lactam D, beta lactam E, this way. We get them and we get their binding energies. From those and comparing their experimental data, we make a window of cutting off. That is used in the docking based virtual screening, where we take the beta lactamase in apo form, which is 2-GDN, have some mistakes. So, we repair it and use it. We had 1 lakh compounds and we are down to around 120. For these 120, we do dynamics. Dynamics followed by binding energy determination. We use two methods. One is thermodynamic integration and another is PBSA GBSA, and then cut them up to 50. So, in summary, we have taken penicillin series, cephalothin series, ceftazidime series and cefoperazone series. We apply all the 3 rationale to modify them, and then we develop the library, which is containing about 1 lakh compound coming from all the 4 series. Then we develop parameter, then we do docking based virtual screening, where we take the already established compounds. That is the advantage of working with the beta lactam drugs, and we develop the cutoff window and we did the screening and we obtained 120 compounds. Then from 120 we do the molecular dynamics along with binding energy. For binding energy, we take thermodynamic integration and PBSA GBSA, and we come to 50. All the 50 compound coming from all the series, penicillin series, cephalothin series, ceftazidime series and cefoperazone series, they are all synthesized. Now, what we have 50 compounds in hand, the real compounds. From our imagination of designing logic, we do the library construction, we do the docking based virtual screening, we do the simulation, binding energy determination and cut off to 50. Then we synthesize them. So, now we have it in hand. So, now we start the project with following objectives. Can Butt-Sus be used to inhibit BLASI? So, BLASI is our target. So, our question is, can the designed compounds which we started from around 1 lakh coming down to 50 would be used to inhibit BLASI? How to screen them based on their biochemical potency? We need to develop an biochemical assay, because once we could get the biochemical potency of a molecule, then only what I forgot to tell you, we have checked the in silico admetox and understood that because we have used the major moiety as the already established beta lactam moieties, all our compounds are non-toxic. And also when we say that we introduce any group, we did not introduce groups which could have toxicity. So, we need biochemical potency. If some of the molecules we have now in hand show biochemical potency, can they also inhibit the growth in vivo in real tuberculosis cells system? And if we are successful, so if Butt-Sus could inhibit, it goes through the biochemical testing, it goes to the in vivo testing, and if successful, as we are drug designer, and if you see the drug designing, it is in cycle, you have to optimize. So, we go to understand the molecular mechanism which will help us to go to the next generation, next round and get more optimized, more potent drugs. So, first we do the cloning, expression and purification of the BLASI enzyme. We make the truncated version, 39 amino acid of the protein was truncated, deleted out, and we clone it in the plasmid PET 24, express in E. coli BL21D3 expression cells. We use histag for primary purification and size exclusion chromatography for secondary purification. We develop or we take the help of kinetic characterization using nitrosephin as a reporter. Nitrosephin is an amazing compound. It is pale yellow in color. In the presence of beta lactamase, it changed the color to deep red. The major advantage here is the target here is the 4-membered ring, and the color development is related to the electronic transition followed by the enzyme attack. So, the enzyme which shows effect on nitrosephin must be attacking the 4-membered ring, and as I have already explained, you know that there is no eukaryotic enzyme or prokaryotic enzyme which are non related to beta lactam which attacks a 4-membered ring. Then we want to go for screening of a series of these 50 compounds which are synthesized. So, we put a criteria, the Ki, the inhibitory constant of the inhibition. So, how the inhibition made? First important question. So, the enzyme is there, nitrosephin is a cephalosporin, which means it could bind to the enzyme. So, nitrosephin, when nitrosephin binds, the yellow color becomes bright red. Now, all those inhibitors would compete with nitrosephin, more the inhibitor would be potent to bind, less color would be generated. So, by calculating the difference in intensity of the red color present here, you could calculate the inhibitory constant on the, generally we do on the Michaelis-Menten, it is little different. I am going to show you the result. 5 compounds we get which are having the Ki less than 10 micromolar. So, this is the first one which is having 7.2 micromolar. This is the second one which is having 1.46. The third one we get 2.76. Fourth one 3.34 and the fifth one which is 0.65. This compound is EC19, and how these values mean, I have given some already established compounds. Clavulanate is a very established mechanism based inhibitor. It have the Ki of 6.3. Phenem 1 and 2 again also designed by me, I have done all the designing, that have 0.8 and 0.2. So, that shows that this is a quite good compound, at least in terms of the designing followed by the biochemical assay. So, we decide to test this one, the fifth one, for in vivo, but I could not control myself to give you a very interesting part. If you remember, in this position we have introduced any and every group. We have introduced amino group, hydroxyl group, only benzene, halides, anything and everything, but surprisingly all the compounds we got, they are all phenyl carboxylic acid. So, that give us a thought that might be this is very important, or made us excited to know about the role of this. So, as I told, once we get the compound selected through the biochemical assay, we go for in vivo testing. In the in vivo testing, what we did, we have the tuberculosis cells and we use combinations. We use ampicillin, clavulanate, and we use ampicillin clavulanate combination, ampicillin EC19 and ampicillin phenem 2. If you see the combination of a beta lactam along with the beta lactamase inhibitor works here. So, we get good data for ampicillin clavulanate complex, ampicillin EC19 complex and ampicillin phenem 2 complex. So, EC19 successfully inhibit M tuberculosis cells, phenem 2 also suggest quite exciting results. So, if you think about the EC19, EC19 was designed, then it comes in silico screening, then it goes biochemical and then biology. So, now we want to look at the molecular mechanism, and for that, as a crystallographer I go for protein crystallography. For protein crystallography there are two steps, like two ways to get the complexes. One, you get the protein crystal in the native form, or you crystallize the protein with the substrate. Interestingly, with a lot of try, I was never able to co-crystallize the drug with the glassy. So, what I did soaking. What soaking means? I get the native crystal and I start soaking the drug EC19. These start working, which was not very easy. If you look at the drug, this is a small molecule, but this is a quite big molecule in terms of small molecule. But at least after a lot of try, I managed to get the crystal, and I show you a comparison of data collection statistics. I know that though I talk about those, but you are not very familiar with that, but something which would be definitely interested you is the ABCs. So, generally when we have the same protein, we get the ABCs quite similar, I would say quite identical. But here if you see, we compare with the apocrystal and the change is quite significant. 43.29 changed to 49.41, 71.41 is 67.43 and 84.69 is 74.63. So, this clearly indicates that there are large movements in the crystal. This is the F0 Fc map of the boronate inhibitor at 2 sigma level, which clearly says the good quality of the structure we have solved. This is the molecular interaction. What we see here, we see molecular interactions from the high resolution crystal structure which we found. So, what was the resolution? You could go and check here, you will get to see this is 2. So, we have collected the crystal as 2 angstrom resolution. So, we see loop movement and all, not going into details, but very interestingly we do not see any significant interaction of the phenyl carboxylic acid. If you look at, this is the phenyl carboxylic acid which we are expecting. So, as I told, in 215 to 230 position we see a significant movement. So, in summary, BATCs were designed as transition state analogues against serine beta lactamases. It could also be used as molecular probes to investigate the structural basis of inhibition. As we know that studying enzyme BLASI would be used as an important drug target, as well as understanding to use this group of drugs against drug resistant strain of tuberculosis. BATCs allow rational inhibitor design strategy coupled with virtual screening successfully implemented in Mycobacterium tuberculosis. This is one of the rare success stories so far against tuberculosis, because drug designing is not a very good thing, very successful thing against tuberculosis. But as I told, we look at them and we see that in all of them there is a carboxylic group in the meta position of the phenyl ring. So, I was baffled, confused, but then when I compared, I see that here there is a phosphate group which is common to any beta lactamase structure of tuberculosis. To my surprise, what I find that when I refine, even if I put the phosphate group, there is still some intensity left. I do not know how much I explain this thing to non-crystallographers, but if you remember when I was explaining Coote, I talked about when you put the electron density, fill the electron density with proper model, the electron density will not show you either red or green. If it shows green or red, that means you have something, like if you have green, you have something you did not model, if you have red, you have modeled it which should not be there. So, here in this position, after I would show you this one here, I am talking about this position around. So, we know that a phosphate is there, but unlike in other structures, I have solved a lot of structures in Mycobacterium tuberculosis blasi with different complex like tebipenem, like NXL 104, which is avibactam, but I have never seen the extra green density. So, I talked with the experts and we apply a different technique. I am not going to the technical glitches, technical details, but what for your understanding, we mask the intensity of the phosphate and then we see, as I told, still the intensity is there and this is good enough for a phenyl carboxylic acid. So, we work on that and we, to our surprise, find the alternative conformation of this phenyl carboxylic acid and we get two very important interactions, actually three important interactions with the carboxylic acid group with arginine 220 and threonine 235, which also make the point clear why the loop was moved. Because of this interaction, the loop was moved. So, we got the alternative interaction and this is extremely critical for next drug designing. So, as I told, our major observation is that in addition to the R1 group, the metabenzoic acid substituent in R2 position is necessary for effective inhibition of blasi, as it provides productive interactions with the carboxylate binding region of the enzyme. Understanding the interaction of phenyl carboxyl moiety might be helpful in future for new generation drug designing, and this is the first description of a BATC or BATSA inhibitor against blasi. EC19 may serve as an important lead compound for the rational design of more potent inhibitors. With the insight obtained by this structure function study, we are confident that further optimizations can be reached. And though I am not going into the detail of next new cycle of synthesis, what we did, we actually mimic the position, the alternative conformer, and we introduce a lot of groups like carboxylate, sulfate, phosphate, all of them along with the other part modification that gives us new libraries which we are currently working. So, a brief description of others, we work with DBO moieties. So, DBOs, the diaza bicyclooctanes are a class of serine beta lactamase inhibitors that use a strained urea moiety, as you see here, as the warhead to react with the active serine residue in the active side of serine beta lactamases. Very interestingly, they are probably the very few non beta lactam working as beta lactamase inhibitor. The first in the class drug avibactam was crystallized and characterized in 2012 by me. Several other recently FDA approved DBOs are there. For example, relebactam is FDA approved and few are in the clinical development, nacobactam, zidebactam, and we worked on them. As I told, the avibactam is also called NXL 104. So, we did the kinetics, we did the mass spectrometry analysis of the time range of the reaction, we get the structure. As I told, this is the first. After that, there are quite few studies with different metal lactamases, but I had solved the first structure. You could see the electron density here. This is the interaction of the avibactam in the beta lactamase moiety active site, and this is the comparison of the beta lactam with NXL 104 and clavulanate, the comparison. We also designed novel penem compound. If you see, this is the meropenem, which is carbapenem. The penems are different, but they show similar stereochemistry, whereas the substitution is C2 substitution. Here the substitution is in other side, that is the difference. So, we design those penem 1 and penem 2. If you see their values, the kinetic values, you see we have determined the kinetic values. I have already shown the in vivo work. We have crystallized penem 2, and very interestingly, if you see, the drug now converted this, there is a change and it converted to a 7-membered ring. It is a unique reaction mechanism, and we propose this as a endotrig cyclization. So, it is extended cyclization making the 7-membered ring. You will clearly see the presence of a beautiful 7-membered ring here. So, what is our hypothesis, that when a bacterial enzyme attacks here, there is a modification, rearrangement of the ring, and a 7-membered ring containing new compound develop. Because of this, up to now there is no report of resistance against this class of novel penems. So, coming to the overall discussion, structure based drug discovery have travelled a long path, and as discussed in this module, that today this field reached in its heydays. It is on the top, a lot of work is going on. With the advancement in so many related paths like structure solution methodologies, next generation sequencing, prediction based modeling, advancement in computer algorithm as well as computer power, all contributing significantly towards advancing this field. But even after all of this advancement, this field still faces a lot of challenges, such as upgrading the efficacy of virtual screening methods, as we have discussed, improving computational chemogenomic studies, boosting the quality and number of computational web sources, improving the structure of multi-target drugs, enhancing the algorithms or toxicity prediction, the ADMETOX, collaborating with other related fields of study for better lead identification and optimization. Computer aided structure based drug discovery is an integral part of multidisciplinary work. Computer aided drug discovery can be used in combination with combinatorial chemistry or high throughput screening by means of various algorithms to prepare combinatorial libraries for high throughput screening, including chemical space characterization. Virtual screening is known to shorten the time and cost of high throughput screening methods. The major drawback of virtual screening is that while generating screening libraries, it ignores the protonation and tautomerism effect as well as ionization states of compounds, thereby missing out on significant hits. Availability of limited experimental data and reliable output of computational methods cause researchers to ignore tautomerization, but they are still irresistible. You have to work on them. In the drug discovery process, ADMET prediction remains a hurdle, as we have discussed in details. Nonetheless, availability of various computational methods for prediction of these values has reduced the time and number of tests on animals. Further development of informatics toxicology is needed. In the de novo lead generation method, though this process seems to be efficient and acceptable, there are limitations of the linking procedures. The first limitation is that the linking fragments should be placed accurately in the cavity for appropriate linking. Moreover, de novo design is thought to be fully automated, but still there is some work to be done manually, which is quite laborious. The sum is not sum, it is a lot. Furthermore, compounds designed by this technique are not always easy to synthesize in the laboratory. Thus, new software is needed that considers the synthesis factors while including de novo designing of compounds. You design it, but you have to think about the possibility of synthesis. That is what is ignored still now. In the case of molecular docking, a variety of docking algorithms and scoring functions are available, but it is important to choose an appropriate scoring function, as I have discussed in details, which require deep knowledge about such software. The limitation of the scoring functions are a major drawback among docking programs, because this software provides an efficient evaluation of ligand binding energy, but ignores the accuracy. Several molecular determinants such as electrostatic interactions and entropy calculations are entirely ignored during ligand binding energy calculation. No single software package is suitable for work with all type of proteins and ligands. Similarly, accurate binding affinity calculation is still debated. Despite a lot of improvements and current development in structure based drug discovery, a consistent solution is yet to be developed. To overcome fundamental issues such as considering water molecules and flexibility of a target molecule, revolutionary innovations are still needed. We have discussed all, but just I have summarized, and now it is time to say thank you to all of you. This is a long course. I have tried my best to tell you the story of structural biology, which includes introduction. I have introduced about how to read the biological molecule, because that is the first requirement. Then I shifted to protein. I talk about protein. I come to different structure solution technique, where I talk about structural biology, high resolution technique like X-ray crystallography, NMR and cryo-electron microscopy. Then I talk about spectroscopy, the low resolution techniques. Then I talk about PDB, visualization, MD simulation, and then I end up with two applications. One is protein engineering, another is drug development. It is my sincere effort, and I hope that especially the young generation who are following this course would get to be interested about this subject, would want to explore the space of structural biology research. There are so many new things happening, especially a lot of interdisciplinary connections are developing, with more and more data science coming, more and more predictive work is going on, more and more sequence every day coming through genome sequencing. It is really the correct time to come into this field, work and innovate. I thank all of you for listening me. I agree that a lot of lectures are long, but that is because I have sincerely tried to include most of the things and make this introductory structural biology course a platform to get an understanding of all the basics and their correlation to the research. I would not take again long time. I will finish here just by thanking all of you, and as I say every time, please feel free to connect us, send us emails with your queries. To finish with, I will talk just two things about the exam. As you are following the course, you will see we have given assignment of 10 questions in each module. So, already you have 120 multiple choice question. The exam will have multiple choice questions like this only. So, you just go through the slides properly, listen to my lectures, and it would not be difficult to answer every questions. I am finishing this course with just one additional information. This course will come again in next January. So, I hope this message you will pass on to your juniors. Thank you very much.